Hi everyone, Azim here, and I'm about to go over the stages of mitosis as shown in cell models. So this model is actually just a regular cell model, and this is the nucleus of the cell. And represented inside of the nucleus is the DNA, the genetic material. It's shown as a bunch of, it's supposed to be helical, but very loosely strewn about stuff within the cell, within the nucleus. It's actually not, it is organized, but not as organized as it could be. So it's very loose, very not as arranged as it could be. Imagine like a bunch of clothes thrown out on the floor, just piles and piles of clothes, and you can't tell one piece of clothing from the next. When DNA is in this form, it's called chromatin. Chromatin with an N at the end, chromatin. So this is a cell in interphase. During interphase, this is when a cell is doing its normal activities, and it could be when it's a cell is preparing for cell division, for mitosis. Mitosis is where a cell will divide either because it needs, an organism needs to grow and develop, or it needs to replace dead tissue. So during interphase, in preparation for mitosis, one thing that needs to happen is we need to duplicate the amount of DNA. So imagine we get 2 copies of this. But now we need to separate it out. Now that we have 2 copies, we need to split it up like a couple breaking up and separating their stuff in their house. So the easiest thing to do is once you've separated it, you need to pack it up. The first step of mitosis, when we're actually starting to divide, is we're packing up our DNA. The DNA is tightly coiled such that you can actually see it under a microscope. When you look at this under a microscope, the chromatin, you don't really see the individual DNA molecules. But in prophase, you can see the individual molecules. You can see what we call now individual chromosomes. Chromosomes. So the chromosomes are tightly coiled. The nucleus goes away because if we want to separate things, separate the DNA, we need to get rid of the nucleus. And you'll have to use your imagination here, it's not shown, but the DNA has been duplicated already during interphase. In prophase, we see that the nucleus is gone, the chromosomes are tightly coiled. Some other things that happen in prophase, not really shown here, but this is another model of prophase. There are spindle fibers coming from centrioles. This is supposed to represent a centriole. Here are spindle fibers. These spindle fibers will attach to either copy. So imagine this is 2 copies. One spindle will attach from this side, from one centriole. Another spindle will attach from this side to over here. Because we're preparing for that splitting up of those copies. So this is still considered prophase. The next step is to line things up. We've gone from prophase to metaphase. In metaphase, the chromosomes are lined up, 1 copy on one side, 1 copy on the other side. So we'll eventually get chromosomes over here, and we'll want chromosomes to come over here. You can see the spindles attached to each copy of the chromosome. This is the beginning of anaphase, the 3rd phase of mitosis. In some textbooks it might say that this is the 4th, but we're going to call it the 3rd. We're starting to see the split between those copies of chromosomes. The spindles are pulling to this side, the spindles are pulling to this side. So here's anaphase a bit more progressed. Starting to pull to either side. The final step of mitosis is telophase. The chromosomes are almost to either side, and we're starting to see what's called a cleavage furrow develop. The membrane is starting to pinch in the middle. The development of a cleavage furrow during telophase, this is called cytokinesis. And here we can see cytokinesis completing. The full split between the cells. So we started with 1 cell during prophase, and we're ending here in telophase, finishing off with 2 separate cells. Now that we've separated the DNA out, we don't need it to be condensed anymore. So the chromosomes can go back to chromatin form. But we have 2 nuclei now because we have 2 distinct cells. And this will eventually split, and we'll have 2 fully unique cells, fully separate cells. They are not quite unique because, at least the plan was genetically, this cell should be genetically identical to this cell. the DNA should be an exact copy, barring some mutations in the process. Again, the end goal of mitosis is to produce new cells that either replace damaged, dead tissue or for regular growth and development.